So hi, my name is Kartike, and I'm going to be presenting my research project, which is high impact genes in nicotine-treated breast cancer cells. So what I'm going to be covering in this presentation, what the objective of this research project was, some background, some of the methods that I used, the results that I got, and a summary, and then finally conclusion. So the objective of the research paper was to identify high impact genes that was related to breast cancer observed in nicotine treated cells, and to find related biological pathways to further investigate cancer progression and to hopefully pave the path for future research. So some background about breast cancer and nicotine and gene expression. So breast cancer accounts for around 1 in 8 cancer diagnoses for women globally, and this often involves altered signaling pathways and gene regulation. And talking about gene regulation, basically what gene regulation is how proteins are produced from genes, and basically like how much protein is produced when they're turned on and off. And the gene regulation and expression can promote or suppress cancer development. Nicotine is a dangerous additive that's used in tobacco products, and evidence suggests that nicotine may enhance tumor progression and metastasis by altering pathways. And some methods that I use is, first, I found a public data set that had nicotine treated breast cancer cells, and I found a velocity ranking for each that was provided in the data set. I used the velocity ranking to filter out the genes that had a valid velocity ranking and a statistically significant p-value, and then also an extreme statistic value. Let me talk a little bit about the velocity ranking. So the RNA velocity estimates the future activity or state of a cell by comparing spliced versus unspliced mRNA. A valid velocity ranking basically means that the gene is actually driving change within the cell and not. And without the valid velocity ranking, you can't trust that the gene is actually doing something in response to the nicotine. And a p-value tests whether the observed difference in gene activity is likely due to chance or is real. Valid p-values indicate strong evidence that nicotine is actually impacting the gene, and this ensures that you're not just picking random fluctuations in gene activity. Genes with extreme changes or high statistical scores are more meaningful and more significant. And these outlier genes are most likely to play a big role in cancer-related pathways. So some results that I found, so these are some pathways that I found that was related to the genes and their functions. So cell cycle regulation, immune system, and cell proliferation. So many genes were found to have an abnormal regulation and a function that was linked to breast cancer. In normal genes, pathways controlling growth, repair, and death are tightly regulated. However, in cancer, these pathways are often overactivated or deregulated, often overactive in breast cancer, and this basically drives cell proliferation or tumor growth. In cancer, irregular proteins can activate the immune response, which is basically which can cause inflammation, and this can cause tumor growth, and also cell cycle regulation. So what can happen in cell cycle regulation is the checkpoints can be either skipped or like not monitored correctly. So a cell that has an abnormal growth or abnormal function is not properly checked by the body and can continue to grow without any control. Yeah, this will basically cause cell proliferation, which is a large amount of cells that are forming a tumor. An example of this is CDK1, which controls the cell cycle and works with cyclin A and B to ensure that division happens only at the right time. But if CDK1 is overexpressed or hyperactivated, cells can bypass normal checkpoints, and this lead to uncontrolled entry into mitosis and further fueling rapid tumor growth. A quick conclusion of what was covered: nicotine is not a direct carcinogen, but it influences cancer progression by modulating gene activity, and this causes long-term changes in cells. Pathways such as cell cycle, proliferation, and immune system responses are all hallmarks of cancer progression. So why is this significant? This basically provides molecular evidence for nicotine's role in breast cancer progression and suggests potential therapeutic targets for intervention. This validates findings in experiment models and also offers information and insights for further researchers or research projects. And this extends analysis to other cancer types other than breast cancer as well, affected by. Yeah, thank you for listening to my presentation. I think it was really good. I learned many tools that I didn't know. Also, how to write the research paper. I did not know that. In terms of mentor feedback, I think my mentor was very, very good.